Good afternoon, everyone. My name is Mark Dirks, and on behalf of ACRL and Choice, I'd like to welcome you to today's program, Librarians Improve Science, Impacting Research Quality Through Transparency and Reproducibility, which is sponsored by JoVE, the Journal of Visual Experimentation. Today's discussion is one in a series of sponsored webinars from ACRL and Choice that addresses new ideas, developments, and products of interest to the academic library community. Free to users, these structured 60-minute live presentations provide the opportunity for interactive discussions of important new issues in academic librarianship by librarians, vendors, authors, and other interested stakeholders. Before we get started, I'd like to point out a few features of the webinar software. In the main area of the screen, you should be able to see the slide decks and follow along with the presentation materials. Along the right-hand side, you should also see a Q&A and a chat panel. If you don't, there are a couple of buttons labeled Chat and Q&A in the upper right-hand corner of the screen, and you can just click those to activate the panels. Please use the Q&A panel to submit questions to our speakers throughout the presentation. At the end, we'll take a few minutes to answer them, so again, please do send them in throughout. And if you're experiencing any technical issues, please use the chat panel to let me know, and I'll troubleshoot the issue with you privately. We are also using the hashtag ACRLChoiceWebinars during the event, so if you have another screen handy, shout out to us. We're at choice_reviews on Twitter. Also note that we are recording today's program, and everyone who registered should receive a follow-up email with a link to the archived version. We will also, in that email, provide a link to the PDF of the slides so that you can get your hands on the references at the end. All right, so our speakers today are Dr. Justin Cherny, Patricia Ayala, and Melissa L. Rethlefsen. Justin obtained an interdisciplinary PhD in biology and computer science from Wesleyan University, which focused primarily on identifying mRNA translation initiation on a transcriptomic scale and the development of novel approaches for quality assessment of peptide searching algorithms on a proteomic scale. During his PhD, he had the opportunity to explore a variety of scientific disciplines, including bioinformatics, molecular biology, genetics, cellular biology, developmental biology, relational databases, algorithms, and a host of wet lab and computational techniques. Justin is continually motivated by JoVE's mission statement, which coincides perfectly with his passion for interdisciplinary work, innovation, and effective integration of new techniques. In his spare time, Justin loves woodworking, hiking, volunteering for manual labor, and firewood acquisition. Patricia is the Research Services Librarian at Gerstein Science Information Center at the University of Toronto. She's co-led the development of the Systematic and Scoping Review Service, SSRS, at Gerstein Science Information Center, focused on collaborating with faculty and researchers engaged in systematic, scoping, and other types of method-driven literature reviews. She established a liaison relationship between Gerstein and the Equator Network with the purpose of developing a network for librarians across the globe and organizing outreach opportunities advocating for the role of librarians in research, proper reporting, and research integrity. She's currently co-leading research on the challenges and opportunities of early career researchers undertaking systematic reviews. As an ambassador for the Open Science Framework, a tool that facilitates integrity and transparency in the entire research cycle, Patricia conducts workshops and teaches how initiatives like this one can be used to align scientific approaches with scientific values. Melissa is Associate Dean, George A. Smathers Libraries, and Fackler Director of the Health Science Center Libraries at the University of Florida in Gainesville, Florida. She led the creation of the first National Conference on Research Reproducibility, Research Reproducibility 2016, hashtag Make Research True, its successor, Research Reproducibility 2018, Building Research Integrity Through Reproducibility. She's received many awards for her work in medical librarianship, including being named as a Library Journal Mover and Shaker in 2009 and receiving the Estelle Broadman Award for Academic Medical Librarian of the Year in 2015. She served as a member of the Medical Library Association Board of Directors from 2015 to 2018 and is currently a member of the Medical Reference Services Quarterly Editorial Board. She's published a book, Internet Cool Tools for Physicians, over 20 peer-reviewed research articles in journals including PLOS ONE, Journal of Clinical Epidemiology, and Mayo Clinic Proceedings, and over 50 non-peer-reviewed articles, including the articles in JAMA and Minnesota Medicine. And with that, we are ready to get started. So I will pass the ball over to Justin.
Hi, everybody. So I just wanted to start kind of with some broad strokes on reproducibility and some of the impacts that it has. So I'm Justin. I'm the VP of Operations at JoVE. I'm essentially this guy with the green test tube, hair color to match, in fact. I personally identify with reproducibility mainly because I was this guy with the green test tube. And basically, during my PhD, my main goal in life was to publish a paper, publish some novel research of some form. And what I did when I started out as a graduate student is I essentially didn't know anything. I didn't know my elbow from my ankle when it came to research. So I tried to read the literature and figure out what I was doing, and then I was going to apply the information that I learned to do new research with the ultimate goal of publishing. And this is the cycle of a scientist, essentially. So the issue that came, though, is that there's this breakdown currently for reproducibility. And we think that this has to do with the way in which things are published and the way in which we can actually talk about and interpret scientific results through methodology and through a bunch of other ways. So what we really were focusing on here is that as reproducibility as an issue is that when you have this decrease in the ability to translate published research into your own research, you decrease knowledge transfer between labs and you don't do productive research. And the thing is that this isn't just a me problem, it's just not me with my green test tube. In fact, reproducibility is actually a very serious problem. In the US only, the NIH funds about $40 billion a year in research funds. So the issue is that a bunch of independent studies came out that suggest that 60% to 90% of the results that are published are not reproducible. That's a big deal. This has huge impacts in society. It delays the development of new medicines. It's bad for institutions in essence. Poor productivity for institutions, less publishing, less recognition. And for scientists, it really makes for very difficult careers. It's actually really hard to be a scientist when you can't get things to work in the lab. So I'll kind of give you an example just to bring you, on the same page with the whole PhD life. So one of the issues here is that if you kind of look at how scientists deal with these issues in the lab is let's take this figure on the left, the one with the subheading Sugihara et al. In 2010. So let's imagine I'm my graduate student self and I'm reading a paper and I want to translate something into the lab. So I'm like, what do I publish a paper on? And I come across this paper and I say, okay, so what this graph is showing me is that on the y-axis you have the DOX accumulation fold in various tissue samples. So basically, I look at this and I say, okay, this DOX chemical is something that's supposed to treat cancer, and then when you include it with this IRDG compound, it targets the tumor. So awesome. Basically, what this means is that I could use this paper to figure out how to selectively target tumors. What a great plan. This is going to be my paper. I'm going to go on and be a famous scientist. I am going to use this information to accomplish my goal. So I take it to the lab and I do it, and it doesn't work. This is the story of a scientist, unfortunately. It never works. It hardly ever works the first time. So I do it again and again. So at this point, I've tried this 3 or 4 times. It still doesn't work. So here's my problem. At this point, I have to make a call. Am I an idiot? And am I bad in the lab? Can I get this to work? Or is there something wrong with the protocol? Maybe I'm not interpreting this correctly. And the thing that's important here is that I am not unique in this scenario. This is not a unique problem. All scientists face this at some point in their careers. So as one of those studies that I mentioned earlier that tried to perform a reproducibility experiment, now these are experts in cancer biology who try and recreate the protocol as it was provided in the original publication, and they find no statistical difference. So what does that mean? They couldn't reproduce it. It didn't work. So at this point in my graduate career, I've now probably invested a couple of months at least, maybe even a year in trying to get this to work, and it doesn't work. So what do? I have to find someone at the institution who's smarter than me, who can help me figure out what I did wrong, who knows how to figure this out. And if I don't have that availability, then I have to actually go and seek out the original authors to be like, what am I screwing up? What's wrong? Because it's very likely that Sugahara et al. Are not lying about this. I'm sure that they were successful in showing this. They clearly have the data to back it up. The problem is that I can't make it happen, and that's a big issue. So that's really what one of the main issues with reproducibility is. And there's a solution to this, though, in that when you're looking at these issues, typically what you get in the text portion of the protocol is this text on the left. Now, you're lucky if you get this much description, and it says, position the metaphase spindle at 3 o'clock and hold it with a holding pipette. Apply piezo pulses to penetrate the zona pellucida. Touch the metaphase plate with the enucleation pipette, aspirate the spindle, and withdraw the pipette. Well, that's all wonderful, but piezo pulses sounds like a yoga move, and the zona pellucida sounds like something that surrounds Area 51. So I have no idea what these things are as a non-stem cell biologist, as a non-embryo biologist. This is actually going to be really hard for me, and I would be lucky to even get this text included in the publication. So how am I possibly supposed to reproduce this work? Now, with this simple image here, this is actually showing you exactly what you need to know. Here's something held at 3 o'clock. Okay, I see there's something holding. It's holding this embryo. I got this other thing. I apply the piezo pulses. If we had the video, I was hoping that I could get this to play, but unfortunately it doesn't look like that's going to work. So I put the pipette directly up to it, and then I puncture into the thing. It says touch through this outer shell. Touch the metaphase plate with the enucleation pipette. Aspirate the spindle and withdraw the pipette. Well, in the video, you would see this light gray shadow is actually the spindle. You pull it out, and you squirt it outside, and you're done. And that's the thing, is that in this case, it's the visualization of the methodology that really makes this useful. It makes it substantial, and it makes it easy to transfer between labs. And that's one of the big things that we have to address in terms of reproducibility. So one of the things about reproducibility in general, and I really like the title of this slide, is that reproducibility has a responsibility. It is all of our responsibility to deal with reproducibility as an issue. We, as JoVE, as publishers, we focus on it. In fact, it's what builds us. It's what supports us. It's what we care about. We help scientists do that. We help peer reviewers who review the scientific content to make sure that the data is reproducible. And one of the things that I've heard a lot throughout the reproducibility conferences I've been to is that there's this big focus on data preservation. And data preservation is really important in terms of reproducibility. But it's not the only thing that's important. Because in that example I showed you before, data preservation isn't going to help me there. What's going to help me there is the methods. And that's the key here, is that methods help reproducibility. You have to do both of these things. And librarians are actually very uniquely situated in this case to really help out, to really be advocates for reproducibility and help address this crisis. So I like this slide a lot because of the image mostly, but librarians are agents of change. And one of the things that librarians can do is to educate and really help faculty and students and management understand what solutions are available to help address this issue. Focus on solutions that increase reproducibility from vendors. There are these opportunities and these tools outside, which we'll talk about a little bit later. And also encourage faculty to improve their publications to increase reproducibility and really take this charge on. So librarians really are kind of at the hub of all of these intersecting pieces and have enormous ability to influence this movement. So with that, I am realizing that I am actually the least important person at this webinar, and without further ado, I'm happy to introduce Melissa to take up this charge and continue talking about reproducibility. Thank you very much.
All right, well, thank you very much, Justin, and I think that Patricia and I wouldn't be here today if we didn't agree that librarians really are excellent agents of change to help solve this problem. But just stepping back very quickly, we didn't want this to be a webinar just defining reproducibility, but just to give you some idea of the kinds of things that we are talking about. Often you'll hear the word reproducibility somewhat interchangeably with replicability or repeatability. Here we're really addressing reproducibility as a sum of all of the different parts that could go into reproducibility, replicability, and repeatability. So that might include things like, again, as Justin mentioned, are the methods transparent and repeatable? Is there enough detail actually provided in the manuscript? Some things that might include would be a data analysis plan, how they are actually using their analysis software and their code, what kind of reagents are being used, what kind of chemicals or biological materials, how are those being validated, how have they been validated. It might include things like needing a full protocol for an experiment or the data itself. And then, of course, there's also the whole component of results reproducibility. So if you're running the same experiment, are you getting the same results? If you have the same data in the code, do you get the same results? If you have the same analysis or reanalysis, are the conclusions the same? There's all sorts of different aspects to this whole problem of research reproducibility. And again, why librarians? You hopefully have already heard a few terms that seem familiar to you or that are even second nature to you, such as data, analysis, software, code, protocols. All of these are concepts that are really becoming more and more commonly used by librarians to describe what we do and what we know. And chances are you're already working on helping to increase research reproducibility. Beyond data and code and data management plans, librarians are experts in the system of scholarly communication in whatever discipline or disciplines that we work in. So adding knowledge of reproducibility and transparency concepts and tools is a really small step towards helping researchers at our institutions, our citizenry, and of course ourselves. Justin mentioned earlier that there is considered a problem around reproducibility. You may also have heard it called the research reproducibility crisis or the reproducibility crisis. So what is this crisis? Essentially, it's been the dawning recognition that perhaps the science that we've been relying on isn't quite so good as we thought. This really started becoming noticed in 2005 when Dr. John Ioannidis, who's now co-leading the Metrics Meta-Research Group, or Research About Research Group at Stanford University, published this landmark paper, Why Most Published Research Findings Are False. This sort of shook up a lot of things, and ever since 2005 when that was published, there have been a lot of large-scale replication studies, a lot of major cases of fraud, and awareness of issues surrounding especially chemical, biological, and instrumental misidentification and miscalibration, all of which have really confirmed the idea that there's a lot of bad, shoddy, or just plain false research that has entered our system of scholarly communication. And this whole concept of crisis has really hit the popular media to the extent that there are actually now journalists out there who are on the reproducibility beat. So now the public is really more and more aware of the errors of science than ever before. Though most researchers, of course, want to do good science and also want to have the public trust their science, it wasn't really until the NIH and other funding agencies started taking notice of this crisis that reproducibility became a really hot topic that our researchers on our institutions wanted to know about. Our researchers need help to deal with all of the new guidelines and things surrounding reproducibility. So why us? We're really good at doing these things. How can we help our researchers, our scientists, even citizen scientists? We have these great skills, and we can help put them to use in promoting reproducibility and maybe even solving the so-called crisis. We're great at building awareness. We're great about teaching people about new ideas, new tools. We can provide tools, and of course, we can always advocate for what we believe in, high quality scholarly communication. And now I'm going to pass it on to Patricia.
Hi, thank you, Melissa and Justin. So, I'm super excited to be here, and I'm going to talk to you a little bit about transparency. So transparency as a principle is nothing new, or it certainly shouldn't be, in the practice of research and science. It is a long recognized principle of responsible research, from disclosure of conflict of interest, peer review, sharing data, and authorship. The idea of transparency is central in demonstrating that research has been conducted responsibly, and so therefore it can be trusted. Now, transparency and openness are components of open science, and open science is really, has this momentum right now as a movement, but it really is, it refers to doing traditional science with more transparency involved at various stages. So, for example, by openly sharing code and data. Many researchers already do this, but don't call it open science. So that all sounds great, and so what's really the problem, right? Like, well, because, you know, as you've heard Justin and Melissa discuss, that hasn't been the case recently or throughout the years about how research is conducted and reported. Typically, what we see is something like this, right? Now, I'm a big Harry Potter fan, and I really wish I had this wand and magically make things happen, but so much of the richness and value of conducting research is about the process. The results really are nilled if they can't stand on the rigor and transparency of the methods in which they were conducted on. So we're really left wondering what happened. You know, many times the end result is also not published, but that doesn't mean that it wasn't valuable. So, you know, again, like, this is what we see typically. So this brings me to the importance of workflow and documentation. As we've heard, a lot of it is obscure. It's hard to reproduce when we don't know how things happen or how they came to be. And just most importantly, you know, we're human. Like, I ask myself that question, like, will I remember in 2 years exactly everything that I did, every choice that I made, if I hadn't documented each and every stage of my workflow properly? Every piece of email that I sent, every data that I collected? And the answer is no. I may not even remember a week after I did those things. So again, transparency is one of these key remedies to the reproducibility crisis, but it also encourages greater responsibility in and for the research stages, throughout all the research cycle. Now, you know, there's initiatives and guidance out there when it comes to declarations, reporting guidelines, manifestos, policies, and frameworks. And I know it can be a little overwhelming to just kind of say, okay, well, I want to be more transparent about my research. Well, there's guidance out there to help you. And, you know, they cover many aspects, metrics, authorship roles, reporting standards for different study types, and research assessments, just to name a few. And while they're influential, what they really need is the adoption and broader acceptance among researchers and stakeholders, and more discussion. And we're part of this because, as Justin said, you know, this crisis is really our crisis. It's everyone's crisis, because research and science, it's all of us. Now, in addition to guidelines, there's also tools that can facilitate being more transparent in every stage of your research process. So that goes, so I took this slide from the Open Science Workshop in Vienna. They did a fantastic job synthesizing a variety of slides, a variety of tools, and by no means this is exhaustive. Since then, it's continued to grow. You can access the website yourself, but there's, you know, there's tools that run the gamut to help you in your workflow in everything from searching, the analysis, the writing, the publication, outreach, even assessment. But I wanted to take some more time to talk about one tool in particular in a little more depth, and that's the Open Science Framework. So when I started, you know, getting the chatter about reproducibility and lack of transparency in research has been there. you know, like a curious librarian and someone who adores her job. Like I ridiculously love my job. I thought, okay, well, let me just take a look at one of these things. So I picked one tool, and I started learning more about it. And I decided to, you know, after I got myself informed, I realized and all the different functionality that it has, and I started with my own actions. So now every single one of my projects, I invite my collaborators, if I have them, to join in. We register our project, we register and report every single one of our steps. OSF is pretty neat in that it allows you to register reports. You can decide how much of your research and your project you make private or public. It allows for wonderful collaboration. It provides individual DOI and identifiers for different components. You can embargo your work. There's also time-stamped on when a project was created, regardless if you made it public or private. And it has this wonderful bilateral functionality among different tools because, you know, like the goal here is to improve openness and reproducibility. And, you know, there are other tools that researchers use as well. So the strategy is to accept that many tools are used and to connect them through one main tool that you use, in this case, the OSF. So by connecting our workflow, we can provide, you know, our audience significant gains in automation, efficiency, and immediate value that reinforces even further changing your behavior about being more transparent. Another neat thing is that it allows you to see the impact and visits to your project. So it's kind of like a, you know, who doesn't like badges and numbers of file downloads? So it's you know, it shouldn't be the incentive or the reward, but, you know, like you're able to see the impact that you're having, and perhaps, you know, the person that visited this was another convert. So the OSF allows you for that. Now, that all sounds wonderful, but with this in mind, you know, like let's talk about some of the barriers and incentives that you're probably thinking about right now. So barriers or incentives. So, you know, some of the barriers that it's not rare to have in your mind right now, to have come by or having discussions, that, you know, like the potential that your work might get scooped. There's sensitive and private data. There's a learning curve to these tools. There's institutional organizational policies that may prevent you from being more transparent. You know, debunking grandfather norms of how currently research is being assessed, right? Impact factor and other current reward incentives, and infrastructural support. So those are just some of the barriers, and by no way I'm dismissing them. And some of the incentives that you hear are like, you know, it's faster dissemination, you get constructive feedback, visibility, you comply with funder requirements, you can, you know, benefit from peer learning. And I think they're so difficult to sometimes, so we know that the incentives are good, and we know that, you know, all of these things foster just better science, better research. But the tricky part is that the goals and rewards of publishing are immediate and concrete, and the rewards of being transparent and getting it right are distal and abstract. You know, we all have beliefs and ideologies and achievement motivations that influence how we interpret the own reporting of our research. And even if we're trying to resist this motivated reasoning, you know, it's just very difficult to detect it among ourselves. So even though I can see those biases or those aspects in other people, it's hard to figure that out on your own. And so there's also, no one is policing you, right? This idea of being transparent really is your own moral imperative. But even if I was able to accept that I have these biases, we're all busy. We have to, you know, like just be able to exist within the limited time that we have. But the one thing I wish that everyone listening would just kind of stop and think about is that how many of these are real, how many of the barriers are real, and how many of them are perceived, and what can we do about our own barriers, about our own biases. So you can just start with one. Start small. Start with yourself. You know, context matters, so we must adopt what helps us, adapt what's necessary, and develop the rest. And the one thing I would encourage you is that you at least consider everything. And remember that we don't exist in a vacuum. Being transparent really is a choice that we all can start making today. It's not really about the technology, which has improved so much in so many different ways that we are now interconnected and can collaborate with other people. But changing science and changing the crisis that we have right now is really about changing behaviors, right? Like I said before, like this really should be your personal choice, your personal moral imperative, because no one is policing you. So with that in mind, I have an action plan ready for you. So get others on board by engaging others in multi-directional dissemination. In the open science day and age, we can't just broadcast. We have to engage in conversation and welcome thoughts and ideas. Share tools, share questions, share resources, Sometimes people are more excited to hear your questions than your, what you think may be your limited knowledge. It's actually not. Use OSF Protocols.io lab notebooks, any tool available to register your project and outline your research plan. And one way you can do it, I'm not trying to deter you from traditional journal publishing, but for example, a journal has limitations in terms of the amount of content you're allowed to submit, but you don't have that kind of limitation in a preprint, for example, or how you archive and register your data. And not only will that make your research more robust, but it's faster dissemination. You can get incredible feedback that will only strengthen your own work. Find the champions, better yet, become one. And the one thing I want to drive home is that, this is going to take time. So take a deep breath, get comfortable, and don't quit. Just keep going at it. So with that in mind, I'm going to hand her over to Melissa now.
All right, well, thank you. Patricia just did a great job in describing how we can create individual change within ourselves, and also by helping our patrons, our colleagues, our users figure out how to use some of these tools on their own. But what about on a larger scale? I'm going to talk specifically about creating institutional change. And the reason why, of course, is because it's an institution-wide problem. Research and transparency are transdisciplinary. They're not limited to a single discipline. They span all disciplines, these issues. And second, of course, many people can create personal change, but really the whole academic system of scholarly communication, of incentive and reward, really helped contribute to creating this problem. And as we know, changing academia one person at a time is hard, if not impossible. So that's where we come in. Librarians, our expertise is transdisciplinary, and we're not afraid of a challenge. So what kinds of things can we do to really help create institutional change around reproducibility? One of the easiest steps might just be to work on a little coalition building. You could host meetings of interested people, get together journal editors on your campus, bring in speakers on this topic that really cut across disciplines, but really start to break down those silos and bring people who are thinking about these issues on transdisciplinary kinds of levels, get together and get outside of their own field of expertise. Another big thing that we can do is purchase tools and make those tools available for our users, as of course, well as teaching how to use them to our users. That's something that we've always bought resources. This is just another new class of resources that's really designed to help research become more reproducible and more transparent. We can also advocate for getting institutional versions of some of these free tools to really increase buy-in across our universities, such as getting an institutional version of OSF or GitHub or some of these other kinds of tools that are out there. But I think one of our biggest roles in creating institutional change is really around the advocacy and education front. We really can be educators in this space. And to give you a little bit more concrete detail about that, I'll go over a case study of what my wonderful colleagues from the Spencer S. Eccles Health Sciences Library and I did when I was at the University of Utah. So in 2015, I had a really excellent, fortuitous meeting with our then Vice President for Research, Dr. Tom Parks, who's pictured there on the side, in which we discovered that he and I really had a mutual interest, which was research reproducibility. I've been looking at research reproducibility issues through this narrow lens in my own research, but he was already thinking of it on a larger scale. What really could the university do to improve research reproducibility across campus and science in general? So shortly after that initial meeting with him, when he proposed that maybe the library could actually take on some of these types of issues, I proposed developing a library-sponsored conference on research reproducibility. Dr. Parks and the rest of the team at the Vice President for Research Office immediately supported this idea, and he suggested a focus on how institutions could support research reproducibility, and even offered up a little bit of sponsorship money from the Vice President for Research Office. He also started up a group on campus that met regularly, that he knew these people were interested in reproducibility issues. And I was able then to use this group and meet with this group and offer suggestions to this group, and they really helped inform the content of the conference going forward. We used another John Ioannidis article called How to Make More Published Research True as the framework for the conference when it was being developed. We also created a hashtag, Make Research True, which is still in use, so feel free to use it, that we used on Twitter to share information about the conference, but also just general information about reproducibility techniques, new research that had been done, and so on. The conference itself took place November 14th and 15th of 2016. The first day was the conference itself, and we included 3 keynote speakers, and we were incredibly fortunate to get Hilda Bastian, who at that time was at the National Library of Medicine working on PubMed Commons, which was a post-publication peer review system. David Moher, who's the author of several reporting guidelines which help researchers structure their publications when they are submitting them for publication, and John Ioannidis, who wrote the articles that I've been mentioning so far. We also had 3 panels, a series of roundtables for group and more engaged discussion, and a poster session. The second day of the conference was actually post-conference workshops using the OSF, or Open Science Framework. Plus, we also were really grateful to have a science journalist, Christy Ashwandon, come in. Christy Ashwandon is from 538, and she wrote the really influential article called Science Isn't Broken, which I would actually say argues that science is broken, but it was fantastic having her there, and we actually partnered with our science journalist group on campus in order to make that possible. For the conference itself, we had 28 poster submissions, the majority of which did come from the University of Utah, but we did advertise the conference broadly, so we got quite a few submissions and attendees from out of state. We had over 200 registrants and over 100 viewers of the live stream, and every school and college at the University of Utah was represented except for 2, which really to us struck home the absolutely interdisciplinary, transdisciplinary nature of this kind of content. Based on our evaluations from the 2016 conference, from which we received a lot of helpful suggestions, our next step was to create a campus-wide research reproducibility coalition. The coalition was designed to bring people from diverse fields together to talk about reproducibility issues. The coalition met quarterly, and the meetings were led by the Eccles Health Sciences Library faculty. At the same time, we actually established a series of weekly talks to give interdisciplinary perspectives on various reproducibility issues. This was also created to keep learning and awareness strong throughout the year and as a way to gear up for our next conference. We called the series Grand Rounds Research Reproducibility, of course in deference to our position as a health sciences library where Grand Rounds is a really common term for a speaker series, but also because the acronym GER really appealed to me. This weekly series brought in speakers from all over campus, and it wasn't limited to the health sciences. We filmed the lectures and put them all up on YouTube as well. We were then fortunate enough to hire a new faculty member, who's pictured here, who focused on research support. And in particular, she was hired to have a major emphasis on research reproducibility topics. And that's Tisha Mitnick there in the green glasses. She was really, as far as we know, the first librarian who was actually hired specifically with a job description to work on reproducibility nationally. And after Tisha arrived, we began planning for our next iteration of the Research Reproducibility Conference in earnest. We received a large grant from the Department of Health and Human Services Office for Research Integrity to support the conference, which was really fortunate. And one of the things that we did to start advertising the conference was speaking regularly at events across our campus. For example, I gave a talk to our Clinical Methods and Research Group, another one to the Research Administrators Network, and then one to the research deans and the deans of the colleges and schools at the Research Leadership Lunch to really hit home how important this topic was at all levels across our institution. We patterned the 2018 Research Reproducibility Conference off of the 2016 conference with one major exception. For 2018, we actually partnered with our Department of Philosophy on campus, which is the first time that our Health Sciences Library had ever partnered with philosophy, so that was a great collaboration. And we created a credit-bearing course that would lead up to the conference and enable attendees to participate more fully in that conference. We actually also offered it for continuing medical education, continuing pharmacy education, and Medical Library Association credit for others who wanted to attend without receiving credit for it. We received a grant from the National Network of Libraries of Medicine Mid-Continental region to support that effort. And we planned the week-long course to incorporate information about the reproducibility crisis generally, but we really wanted to make sure that this was something where people could really take away concrete, hands-on types of skills. So we included afternoon sessions taught by Vicki Steves, who's at NYU, to introduce learners to practical tools that could better enable reproducibility practices. The course took place June 11th through 15th with the June 15th Research Reproducibility 2018 conference, a required component of the course. The conference had two keynote speakers, Victoria Stodden and Ivan Oransky. Victoria Stodden, who's a statistician who works at the iSchool at the University of Illinois, and Ivan Oransky, who was the co-founder of Retraction Watch. We also had 3 panels and another poster session. We had 143 registrants and 18 posters. It was a somewhat smaller cohort than our first conference, we suspect probably due to the time of the year. But we still had attendees from as far away as Maryland. Evaluations so far have been very positive, but for me, truly the best part was seeing the undergraduate students who attended, particularly those who were presenting in the poster session their own replication studies that they had undertaken. So this slide just reviews what our year of academic effort, our academic year effort was in this area from August of 2017 to June 15th of this year. We ended up having 381 subscribers on our mailing list that we used to send out information about reproducibility themed events. We had 27 lectures, not including the conference or the affiliated course, more than 40 coalition members, and nearly 3,000 video views just for our Grand Rounds Research Reproducibility lectures. We also have now added our full conference feed to YouTube, plus videos from the workshops, which all are receiving a lot of views. I think really, though, the most important thing to take away from this case study is that the Eccles Health Sciences Library and our faculty working on the project became known throughout our campus as the experts on this topic. It opened a lot of doors for us to have deeper engagement in the research practices of the institution. The case study I've gone over took place over about 2.5 years of work, and I will be the first to say that not much has really changed yet. This is a hugely wicked problem, research reproducibility, just due to the sheer complexity of what reproducibility is and what it means down to the tiniest of details. There's actually one paper out there that found that the difference between shaking and stirring a tissue sample made a whole experiment non-reproducible. So it's that tiny. But the complexity is also compounded by the fact that academics are really strongly incentivized to publish constantly, which leads to sloppy research. It's likely going to really require major reviews to tenure and promotion guidelines and revisions, as well as revisions to the things that we value as institutions before real change is going to be realized in this area. Of course, one of the other issues is there are a lot of people out there who think this is not a problem. But if you talk to undergraduate and graduate students, they'll let you know otherwise. They're very concerned about these issues. One of the biggest challenges is working within institutional policies and state laws, which Patricia mentioned earlier, but especially laws relating to expert controls. How does your institution feel about open science and data sharing in light of technology transfer guidelines and export control? It's a challenge for some institutions, and it's something that you'll want to look into if you're not familiar with those ideas and concepts. But of course, there's always that money issue. Who's going to pay for the time it takes to do better documentation? Who's going to pay for the tools that researchers can use to make their research more reproducible? Who's going to pay even to have their cell lines authenticated so we can tell whether or not it's a breast cancer cell line or a cell line from another species entirely? And who's going to pay for our researchers' time? These are huge challenges, and it's going to take years and years of work to help fix them. But we can do our part to help the systems we work within improve, as long as we keep reproducibility in mind. And now on to some of our takeaways from the session before we open it up for questions.
Yeah, so I'll jump in here first. Justin speaking again, for reproducibility being everyone's responsibility. I think this really is true, that this really falls on everyone. I think that publishers, I think scientists, I think researchers, I think peer reviewers, I think government, I think librarians especially, are key movers in this argument. And I think that the focus on reproducibility has to be twofold. I think the data preservation that we've spoken about in the past is incredibly important, but we also need to focus on the methods, because without focusing on how to transfer that technology between labs, you really can't address reproducibility as a whole. And one of the things that's really important about that with methods in general is that science is hard. It's really complex. And one of the things that's really important there is that video publications can really help and can really help transfer that information between labs. And one of the things in general, just if you think about this, right? If you have a simple problem, like you have to learn how to fillet a fish, right? What's your first action? You go on the internet and you type in how to fillet a fish, and you watch a guy on a boat with no shoes and he fillets the fish, and then you go to your kitchen and you stand there with your knife and your fish and you go, yeah, I got this. That's what reproducibility is in essence, and we can help with that. We can address that issue across academia through video publication, through utilizing these other resources that are out there for reproducibility. And I think it's really important that everyone kind of pick this up and move forward with it.
So my two takeaways would be Johnny Cash, walk the line, change your behaviors. Changing behaviors is difficult, and we must start with ourselves and be relentless. Don't focus so much on the tools. Yes, all of these tools are wonderful, and if you think about it, the creation of these tools and principles and frameworks came because there had to be, something that needed to change, right? Like the creation of them in and of themselves speaks to something. But now it's about changing the behavior of ourselves, of the clients and patrons and students and researchers and clinicians and other colleagues that we interact with, and spreading the word in every way we can. If you think about it, the tool is as relevant to, it's as relevant to the behavior in this way. So, toothpaste. They all kind of sort of work, but the point is they only work if you use it every day. I doubt that the latest, Crest flavor is going to make my teeth sparkling white. It's the fact that I brush my teeth daily. So yes, I did go there with that example. And with that, I will turn it over to Melissa.
Thanks, Patricia. And let's all have sparkling white teeth now. That would be also good. But my takeaways are also pretty similar. And I think the one thing that is the most important for people who are on this webinar to take away is to know that this is something where librarians have expertise already. We know about scholarly communication systems. We know about how publishing works, and we can really step in there and help influence some of these things. We can create change, and we can do it together. We can do it at our institutions. We can do it personally. You don't have to be quite as hardcore as Patricia or I, but really small actions count. Even just teaching a few grad students about what OSF is, hosting a workshop on your campus, talking to some people who might have questions about how to interpret the NIH guidelines, passing someone on to one of the National Science Foundation's reports on reproducibility. All of these small actions are going to count in the long-term goal, which really is to change academic culture. So I know that we have several slides of resources that we're not going to go over here right now, but they will be available to you when you are able to access the slide decks when they are published online.
All right, great. Thank you, Melissa, and thank you, Patricia and Justin as well. This is Mark from ACRL and Choice. We have about 10 minutes here for questions, and it looks like we've got a few of them in the hopper already. But I would encourage everyone out there, if you have questions for our speakers today, drop them in the Q&A box, and we will get to as many as we have time for. I would also mention, as has been mentioned perhaps a few times already, that we will be publishing the slide deck on our website, Choice 360, where you go to register for the webinar. You should be able to find it there shortly after the presentation today. So looking through some of the questions here, we've we've got a question from Tess, which is very specific, and I'm not sure if we can say specifically what it was, but Tess is curious about which study, what the study was, where it made a difference, whether the sample was shaken or stirred. Do you remember precisely what that was, Melissa?
That's a great question. In the resources page, the article that is by me, it's called Building Capacity to Make Research True. I cite it in there. So I would go and take a look at that bibliography. I don't quite remember what it is off the top of my head, but if you go to that article, it does mention it in there. I particularly liked that example because I'm a big James Bond fan. So shaken, stirred, totally affecting reproducibility, and your martini.
Great, great. We have a sort of a suggestion here from Hindishi, who says, why not have research articles have an additional mandatory section, recipe, which is really how to reproduce the results. What do you think of that sort of an idea? Or is that really what the methods section really should be doing, but it isn't capturing that yet?
So that's one of the issues, I think, in current academic publishing, is that there's not enough attention given to methods. And a lot of the academic institution is focused on results. And results are very important. It's why we do the science. But the issue is that most of the time, the methods sections read something like the following. It'll say something like, you know, extract tissue from an organism, and then it'll cite references 16 through 19. And then you have to go look at those 3 references, and nowhere in there is it actually described. So I think that it's a great idea to put more focus on having publishers try and do these things, but a lot of them aren't actually equipped to do so. That's one of the advantages for JoVE, is that it is focused on just the methods. The data and representative data is important, but it's the methods that matter. That section is one of the most important parts, I would argue, of a publication, and I think that it's enormously lacking in a lot of places.
I would agree with that as well. I think that ideally that all research articles would in fact have that recipe on how to reproduce the results. But I think part of the problem is that not everyone even knows what kinds of details to put in on how to reproduce the results, and a lot of it has to do with even the software that you're using for the analysis. So it's not just a matter of having a recipe, it's a matter of having your software, your coding, and your data actually available so that people could actually reproduce the results.
And do you have anything to add, Patricia, or shall we move on?
No, I think my two wonderful colleagues have covered that pretty well.
All right, great. We have a question here from, or sort of a statement again from Fred that says the programs that were described, despite their tremendous appeal, are very expensive to facilitate. They take time and money, and for libraries with increasingly strained budgets, they can find it difficult to fund even small parts of these sorts of things. I think perhaps like the conference that you were talking about, Melissa. But I would ask, in response to that, what even for an institution with a strained budget, what are some of the things that you think a library or an individual librarian can do? And I think, Patricia, your whole section was really about that. Would you offer any response to Fred there?
For sure, and I understand, and I agree with you. It's tremendously appealing, but it's expensive to facilitate in many resources, so human capacity, time, all of those things. Like I said, there's things that we can do, even if it's at your individual level, and just sharing knowledge about these tools, or starting that conversation, or whenever you talk to other researchers, or talk to students, or talk to other colleagues and peers, and there's more people out there that are in similar situations as well with constrained time and resources, that, it's kind of like, what is it, necessity is the mother of creativity, and I don't mean that like in a very light response way, but it's true. So you remember the other thing too, is that we don't exist in a vacuum. We don't operate in a vacuum. So there's a whole community out there that can certainly understand those concerns and will also offer some solutions. The OSF tool, for example, that's free. It's completely free. A lot of these tools actually are free and openly available everywhere.
Great, great. And that's great to know that there are, in fact, a bunch of free resources out there that we can use. We've got a few more questions here. It looks like Bill has the question where he says, research pre-registration is usually lumped in with open science, but does it need to be open? A lot of researchers are concerned about being scooped or embarrassed if they share what they've found too early. Is there a way to keep the protocol secure and private until publication, or would that defeat the purpose? What do you think about that?
So yes, oh, sorry.
No, you go ahead.
Yes, you can keep your protocol as private as possible, but if you think about it, I mean, you have to have it complete. With regards to any potential scoopers, the fact that you already have your plan, which is essentially a protocol, is a research plan, you have it already put together, that tells me that you're pretty ahead of the game. If they're too lazy to actually do the work, the fact that they will actually go ahead and scoop you, it's okay, unless you're really, they're really driven into scooping other people. And like I said before, you can register your report. You just don't have to make it public necessarily, or you can create, there's a timestamp. At least I'm talking about OSF right now. There's a timestamp to that. With regards to, if it's too early that will be embarrassed, I think I understand that as a researcher myself too. To put yourself out there is hard, but I think we also have to trust a little bit in that people are actually going to be constructive if they have any feedback to provide to you. And that really is the only chance before your project is submitted for publication to get feedback from people that you may, not otherwise have had the chance to do so. And big teams, I know Melissa talked about or mentioned David Moore, his entire teams when they're doing projects, their projects are also in OSF, and they've actually said that they welcome and they've gotten really good feedback that otherwise they would have missed. So again, there's always pros and cons, but it's a matter of, like really believing, and that's a personal decision that you have to make, whether the incentives or the cons are going to outweigh, the pros are going to outweigh the cons.
All right, and we're running a little low on time, and we've got a few more questions here. So we'll get to as many of them as we can. I think we'll take the one from Hyojin, who says, do you know of reproducibility tools that are well received among computer scientists? And most computer scientists refuse to use open tools like Jupyter Notebook, and they don't really acknowledge that there's a problem with their workflow. Do you have anything specifically for computer scientists, or?
Sure.
Yeah.
Actually, computer science, some branches of computer science are really ahead of this game. So for example, software engineering is pretty on top of these sorts of things. But Jupyter Notebook is certainly one of them. GitHub is a hugely important tool for computer scientists. But there are also other things out there for computer scientists, like Cloud Lab, for example, which allow you to go on and run experiments in an online system that you can use to try and duplicate someone else's experiment or try and do a replication. There are a lot of things out there in the computer science. Some branches are actually now rewarding people for making their code and their artifacts, or like their software, for example, available as part of submissions. So they're actually, I think, a little bit on top of the game. But Jupyter Notebook, GitHub, things like Cloud Lab, I think are really good tools.
CodeOcean as well. It's a cloud-based computational reproducibility platform for code.
All right, great. And we'll take, I think, just one last question here from Fred, who says, partnering and collaborating with library groups, especially groups like ALA, ACRL, STS, SLA, and MLA, is great, but he'd like to see some of the biomedical and biological societies and associations join in planning these sorts of programs. Do any of you have any suggestions along that front about integrating biological and biomedical societies or other sorts of groups with perhaps library groups and other folks?
Well, I think that's a really great suggestion, and I think one that we should probably take into consideration on all levels, because this really is a topic that affects everyone. I think there's a lot of, especially computer science and statistical groups out there that are looking at these and have a lot of themed programs around these topics, but it's still pretty niche in a lot of different areas, like biological and biomedical societies. But I think one of the ways that we can get organizations and associations involved is by having things like the Research Reproducibility Conference, having things like Grand Rounds Research Reproducibility, having coalitions on our campuses, just meeting groups, people who are interested in these topics, because when we're bringing in researchers, scientists, faculty members, students from all of these disciplines, then it can help filter out to some of those other areas that then we can really start to create those kinds of connections and networks with.
Great. All right, well, I think we've come just to the end of our time here. So I will say thank you, Justin, and thank you, Patricia and Melissa, for taking the time to present today. I think it was a really interesting presentation, and I think there are a lot of takeaways for all of our listeners today. I'd also take a moment to remind everyone out there that we did record today's program, so be on the lookout for a follow-up email from ACRL and Choice with a link to the recording as well as a link to a PDF of the slides. I would also mention that you should see a link to our brief 5-question survey in the chat box. If you could take a minute to fill that out and let us know how we did, we would really appreciate it. Thanks again to everyone out there for listening in today and for participating. I hope you enjoyed the session, and I hope the rest of your day is great.